Hereby, I open this academic ceremony at Maastricht University. First of all, I welcome Mrs. Jingnan Huang, and she will defend for a double degree of Maastricht University in the Netherlands and the Universidad de Santiago de Compostela in Spain. And her thesis is entitled Platelet Proteomic Progress and Retraining Mechanisms in Glycoprotein VI-mediated thrombus formation. And I will welcome all the members of the degree committee, and in particular the 4 supervisors. And the supervisors are Professor Johan Heemskerk. He is professor of cell biochemistry of thrombosis and hemostasis at Maastricht University. Professor Albert Sigman, he is professor of protein analytics of the Leibniz Institute for Analytische Wissenschaften, ISAS, in Dortmund, Germany. And the 3rd supervisor is Dr. Garcia, and he's leader of platelet proteomic group of CIMUS at the University of Santiago de Compostela in Spain. And the 4th supervisor is Professor Hugo ten Kate. He's professor of clinical thrombosis and hemostasis of Maastricht University. I will introduce the 6 opponents later, and 1 opponent is not present due to an accident, so that's an unfortunately accident. And his questions will be asked by the chair of the assessment committee. And I will introduce the opponents later during the opposition. Welcome to the followers of the live stream in China, in Spain, in Germany, and the Netherlands, and wherever you are. Mrs. Jingnan Huang, may I invite you to give a summary of your thesis? The floor is yours.
Yes, thank you very much for your words. Then I will share my screen. Okay. Dear projector, dear members of Corona, dear family, friends, and audience, in next 50 minutes, I will give a brief introduction of my thesis with the title of Platelet Proteomic Progress and the retraining mechanisms in glycoprotein 6-mediated thrombus formation. So first, we will start with the introduction. Platelets are the smallest blood cells being released from megakaryocytes. Upon vascular injury, platelets encounter exposed collagen, and the binding of the collagen to platelet membrane receptor GP6 trigger a strongly platelet activation to secrete the secondary mediators like the thromboxane A2, ADP, and also active the integrin alpha-2 beta-3 to stimulate more platelets and amplifying the thrombus formation process. Since we know the important role of the platelets in the thrombus formation, we have 2 questions here. So the first question is, which platelet proteins involved in the thrombus formation? The second question is, which signaling pathways involved in the GP6 and integrin alpha-2 beta-3 mediated thrombus formation? For the proteins, in 2012, workers identified more than 3,000 human platelet proteins with copy numbers. 2 years later, Zella quantified more proteins in mouse, and these proteins including the receptors like GP6 and integrin alpha-2 beta-3, and also the signaling proteins like the PTK2, CRB1, and SHP1 and 2. But when we compare the identified proteins with the platelet transcriptome database, we noticed that there are around 10,000 proteins are missing. So we would like to know which proteins are missing and what are the functions of the missing proteins. For the signaling pathways, there are a lot of research that has been focused on the GP6 and the integrin alpha-2 beta-3, but there are still many downstream signalings are still unclear. So in this thesis, we focused on the two pathways. The first one is CRB1 and the PKP2, and the second one is the SHP1 and the SHP2. But because due to the time limitation, I only give the detailed information about the signal pathways of the CRB1 and the PKP2. According to the background, we have three aims in this thesis. The first one is to review the composition of the complete human platelet proteome and the transcriptome, and then to compare the human and mouse proteome and the transcriptome for GP6 and the GP6-related signaling proteins. Finally, to elucidate the novel pathways of GP6 and integrin alpha-B beta-3-induced platelet activation and thrombus formation. So in the first part, we'd like to solve the first questions. In this part, we make a summary of 67 publications related to the platelet proteomics since 2010. And we found that the identified platelet protein numbers at the range of 2,000 and to 6,000. In our group, we combined all the data from our group and get a database containing 5,050 unique platelet proteins. And in these publications, we also interested with the signaling pathways, and we found that around 50% of the publications were focused on the GP6-related signaling pathways. In order to get the better understanding of the human platelet proteome, we collect the platelet transcriptome database and also established a unique platelet proteome database. For the transcriptome database, we get the data from the blueprint database, and which contain around 20,000 transcripts. And for the platelet proteome, we combined all the publications from ESAs and get a database which contain around 5,200 unique platelet proteins. Well, and then we in the around 20,000 transcripts, there are around 15,000 protein coding transcripts. But in our database, only 5,000 proteins are corresponding to the protein coding transcripts. That's when we combine these two data together, we noticed that there are around 10,000 proteins are missing here. So we would like to know why these proteins are missing and what are the functions of these proteins. To know the functions of the missing proteins, we divided all the protein coding transcripts and the proteins into 21 function classes based on their locations and the functions. And we found that the majority of the missing proteins are the nuclear proteins and the proteins which involved in the transcription and the translation, and also the proteins in also the membrane receptors and the signaling proteins are missing a lot. To get the better database, we performed newly experiments with 30 subjects and identified more platelet proteins, but the membrane receptors and the signaling proteins are still missing a lot. Then we combined the transcriptome and the proteome together to solve these questions and found that the transcriptome and the correlation between platelet transcriptome and the proteome are very bad. But in these triangular patterns, we get a conclusion that the high copy numbers could correspond to the high mRNA levels, but not vice versa. So in this part, we get a conclusion and find the three restricting factors for the missing proteins. there are the nuclear proteins, nuclear locations, and the low transcription levels and the low translation levels. Then we go to the chapter 2. For the chapter 2, we collect all the database. We collect the human and the mouse platelet transcriptome database data and the protein data. When we compare the transcriptome of the human and the mouse platelet, we find that the correlation between human and mouse platelet transcriptome are relatively low here, but the overlap between the human and the mouse transcripts are high. This is also for the protein. When we look at the membrane receptors and the signaling proteins, we were interested and find that the similar conclusions with the genome-wide transcripts and in the membrane receptors and the signaling proteins, we were interested on the several receptors and the signaling proteins. And in these proteins, we also find that both of them are identified in all of them can be identified in both human and the mouse, but the expression levels are different. To get the further research of the signaling pathways, we performed the experiment and flow. In this part, first we collected the blood from donors and then incubated the inhibitor with the whole blood. And after incubation, we pumped the blood flow over the surface of the organist and then observed the thrombus formation by the microscope. So in this part, we demonstrated that the block of PTK2 and the block of CIB1 could reduce the GP6 induced thrombus formation. So in this thesis, we get three conclusions here. The first is we established the new human platelet proteome, which consisted of 5,500 unique proteins out of the total of around 15,000 protein coding transcripts. And we also found that the human and the mouse platelet transcriptome and the proteome are qualitatively similar, but with limited correlations in expression levels. This is also for the receptors and also for the signaling proteins. And we also demonstrated the GP6 in the GP6 and the integrin alpha-2b beta-3 mediated platelet activation and the thrombus formation. The signaling via CRB1 and PTK2 plays an important role. Finally, I would like to thank all my supervisors and all colleagues from ESAS and the universities of Maastricht and Santiago de Compostela. And I also would like to thank all my collaborators from the universities of Cambridge and Utah. And finally, I would like to thank the TAPAS for the funding. Thank you for your attention, and then I will give the time back to the projector.
Thank you very much, Mrs. Huang, for your clear presentation. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Edwin Mariman. He is a professor of functional genetics at Maastricht University, and he is also secretary of this degree committee. Professor Mariman, the floor is yours.
Thank you very much, Mr. Pro-Rector. Dear candidate, first of all, my compliments for the contents of your thesis, and that is also, of course, to not only to you, but also to your promotion team. I think it's a real contribution to the science of thrombus formation. And for my questions, yeah, I was first intrigued by your work on the proteome and the transcriptome of the platelets. But finally, I decided to move to a more chapter with a more functional aspect, and that is chapter 8. So I want to ask you a few questions about chapter 8. And that is the chapter where you study the GPR56 receptor. And you design some or you select some peptides to influence the function. And one of these peptides is PGPR.
Yeah.
And as I read your chapter, I had the impression that you selected it as a possible activator of GPR, but in your experiments, you find out actually that it's inhibiting the activity. So can you comment on that? Did you make a wrong decision there, or why did you think that it's an activator?
Okay, highly esteemed opponents, thank you very much for your questions. Yeah, this question is also confused us before, because at the beginning we just mimic this peptide and wanted to see the activation of the GPR56, but we see the inhibition, probably because that this peptide can because we mimic this peptide first the GPR56 can be self-activated by at the high share rate. So we just mimic the peptide at the as the ligand of itself. But the mimic peptide can't active the GPR56, probably due to that this peptide occupy the binding site, so its own ligand can't combine with the GPR56. I think probably that's the reasons, because we find this peptide based on the publications, but there are only one publications about this peptide. Okay. So probably I don't know, but probably it can't work as it's it should be.
So I saw that the company Sigma sells this peptide also, and it says it's a potent and selective agonist of this GPR. What would you like to say to this Sigma company now you have done your experiments?
Sorry, can you repeat your questions? I'm sorry.
Yeah, the company Sigma sells this peptide, and they call it a potent and selective agonist, so an activator of GPR56. So my question is, what would you like to say to the company now that you have done your experiments?
Yeah, to be honest, we also bought the peptides from Sigma. Yeah, because at the beginning, we found that the results are different with what we supposed. So we bought the peptide from Sigma and repeated the experiment and found the same conclusion. So I don't know, probably we need to do more experiment to confirm this conclusion. Yeah,
but maybe Sigma should be also aware that it could be different from what they state. Okay.
Yeah, probably.
But for CIB, you also designed you selected these peptides, and one was a mutated peptide, which you expected because of its low binding energy to work better, but it didn't. Yeah. Can you briefly comment on that also?
There are so many reasons that we designed this peptide, and hope this peptide can be work better, because we hope that this peptide can go to the platelets better. But we didn't see this. We didn't find it works. Probably because that we changed some amino acid, and this amino acid works in the real world. Because we designed it by computer, but probably this peptide can't work in the real world.
Could it in vivo be a post-translational modification of this peptide, or do you look into that? It's not the case?
I think that's probably because we can't. To be honest, we can't answer these questions. So I think it probably is not important, because because the vet peptides are worked. So
Yeah.
probably the modified peptides is
So,
not important as we suppose.
right, I understand you're not worried about that so much. I have one small practical question. Reading through that chapter, you use a collagen 1, collagen 3, and collagen 4 for thrombus formation. But collagen 1 is from the horse, and collagen 3 and 4 are from human. Why is collagen 1 from horse and not from human?
Because
and could that have an influence on your results?
No, I don't think so, because this is the collagen 1 is the normal collagens we used, and I mean it's a typical collagen. That's why we choose this one. But we also I didn't do it, but my colleagues also tested it. They found that there's no influence.
Yes, a very small question then, just because I was curious. This chapter is mainly about your study of GPR 56, but you don't mention it in the title of this manuscript, and also not in your presentation that you give. Why is that?
Because that's I think that the CIB1 and the PTK2 more important, and because I only have 50 minutes, so I just present the important things that I think is good.
Okay, thank you very much for your answers, and I give the word back to the pro-rector.
Thank you so much for your questions.
Thank you very much, Professor Mariman. The opposition will be continued by Professor Monica Stoll. She is Professor of Genetic Epidemiology and Statistical Genetics at Maastricht University. Professor Stoll.
Thank you very much. First of all, let me congratulate to this very comprehensive thesis.
Thank you.
With all these data that you had to go through and come up with your conclusions. So I was very impressed. I would also like to extend my congratulations to your promotion team. I would like to start my discussion with you on chapter 3, where you compare the transcriptomes and proteomes of megakaryocytes versus platelets. And on page 97, you state that there's a weak correlation between the protein and transcript levels in the megakaryocytes and the platelets. And you conclude that this provides insights on platelet shedding. Could you elaborate on that conclusion a bit?
Okay. Highly esteemed opponent, thank you very much for your questions. Yes. In this part, we found that the correlations between the transcriptome and the proteome are very bad. But we found the high correlation between the megakaryocytes and also the megakaryocytes and the platelets. That means that the majority of the transcripts in platelets are from the megakaryocytes. And then we found that the low correlation between the proteome and the transcriptome, probably due to the locations of the proteins, like the nuclear proteins, because there is no nuclear in platelets, and the nuclear proteins are from the megakaryocytes. Probably that's the reasons, and we also found that the transcription levels and the translation levels could affect the platelet, the proteins. So we think that probably that there are so many reasons that could there are so many
It's okay.
Help us to identify the proteins.
So, dear candidate, could be there another answer to this question? Because you say in your transcriptome, you do not only measure protein coding RNAs, but also other RNA species like non-coding RNAs. And could they contribute to this difference you observe between the two different cell types?
Yes, we detected the mRNA, the protein coding RNAs, mRNAs, and the procedural genes, and the RNA genes, and we chose the protein coding transcripts here because we want to compare it with the proteome. But the procedural genes and the RNA genes are works in other aspects, like that procedural genes can be biomarkers for the cancer, and the mRNA, the RNA genes could regulate the transcription of the mRNAs.
Yeah, very well. So you know, when you write in your thesis, you say you have the high abundance transcripts, which you almost always find back in your proteome, and the low abundance RNAs you rarely find back in your proteomes. And some explanation you give is the sensitivity and the specificity of the assay. But have you ever heard about long non-coding RNAs?
Yes, I heard it, and I just classified these transcripts into the RNA genes. But I'm sorry.
Just one question. So long non-coding RNAs, they are low abundant, so they are very hard to measure, and they are not transcribed into proteins largely. However, some of them give rise to micropeptides. And out of curiosity, because I'm not a proteome person, can you measure micropeptides with proteomics with the methodologies that you used?
I think we can, but probably we need to do some special treatment, like to do some enrichment, because you know this these transcripts are have very low expression levels, so that means it's hardly to translate to proteins, and if they can translate to proteins, probably these proteins with relatively low abundance, it make the low abundance is for the technologies we have yet. It's so hard to identify the low abundant proteins because we use the accreditation method that only select the high abundant proteins. So it's so hard, but we can just to do enrichment, like to remove the high abundant proteins and just enrich the low abundant proteins. That can increase the chance to identify these proteins, I think.
Thank you very much. My questions are all answered. I yield back to the prorector.
Thank you.
Thank you, Professor Stoll. Well, as I said before, the next opponent would have been Dr. Alvarez Castro, and he is Assistant Professor of Pharmacology, Pharmacy and Pharmaceutical Technology of the University of Santiago de Compostela. But unfortunately, he has got an accident and he was not able to join the degree committee. And Professor Mariman, as chair of the assessment committee, will ask the questions of Dr. Alvarez Castro. Professor Mariman.
Thank you again, Mr. Prorector. Dear candidates, Dr. Alvarez also sent me his congratulations on your work. So he's pleased about that. And he sent me a few questions. The first is about something that you also showed in your introduction. He wonders whether the use of the mice proteome in comparison to the human proteome will be beneficial, or maybe it will introduce more uncertainties than that it will help you. Can you comment on that? Why did you move on to the mouse proteome, and how did it help you?
Okay, thank you. Hi, esteemed opponent. Thank you for your questions. In this thesis, yes, we also collected the human platelets, transcriptome data and the proteome data, because that the human is the best models for the research, because it has the similar genomic, it's similar with human in genomic levels, and of course, it's a very good model for the drug development. So yeah, it means that we can find a lot of we can do a lot of researches on the mouse that we can't do in humans, because so like that we can just if we wanted to know some roles of the roles of some receptors, we can do we can just do the gene mutation of in mouse. And yes, if we want to do some researches about the disease, yeah, we can also use the mouse models. That's why we wanted to expand our research on the mouse.
Yes, yes, of course. But we know also that the mouse has a different metabolism and different physiology from humans. So wouldn't it be, should we not be very careful with the interpretation of the data or the extrapolation of the data?
Yes, yeah, to be honest, we cared about this, but yeah, like that we collect the mouse data with the sepsis, and that we found that in the transcription levels, in the genome-wide transcription levels, we found the difference between the healthy and the patients are very small. That means probably there are less influence on it. Yeah, and we also found that the overlap between human and the mouse is high. So probably I think that it can be.
Yeah, so you expect it's very conserved processes in mouse and human.
Yeah.
Okay, thank you. The second question from Dr. Alvarez is on SHP1 and SHP2 signaling.
Yeah.
When PY3K is low, it can regulate GP6-induced platelet response. And Dr. Alvarez asked, how can it be that it's without PLC phosphorylation and without affecting intracellular calcium?
Okay, yeah. Yes, to be honest, yeah, I did this experiment, but yeah, I did this experiment later, before after the thesis, and we found that the SHP1 and the SHP2 have different roles. Normally, the SHP1 could be considered as the positive regulators of GP6, and the SHP2 can be the negative negative regulators of the GP6. So in the aggregation experiment, we found that the SHP2 has the could affect the aggregation more, but in the signaling pathways that we found that SHP1 works more. So we found that the combined of the when we inhibit both the PI3K and the SHP1, we find the decrease of the phosphorylation of the PI3-gamma2, and we also, but for the calcium results, we find that the SHP2 works more, because we also did the dose response of the CRP for the calcium release, and we find the increase. We find that the increase of the calcium release when we use the inhibitors.
Okay, thank you. It's clear.
Thank you.
I have a third question, and that is about the role of the flow of the shear. I have an own question, because shear is apparently very important, and it increases when the vessel is already forming a thrombus and is closing, then the shear will increase. Is it not counterintuitive that there is a system in the body that responds to the increased shear with thrombus formation, because that would aggravate the process?
Yeah. Yes, of course, the shear is very important. Like that, there are some. We can take the chapter 8 for the example. We find that the block of the CRB1 and also the CRB2, no, not CRB2, PTK2, it works better at high shear rate, like the arterial shear rate, but it works worse in the at the low shear rate.
Yes.
So that means,
yeah. So is it not a disadvantage when you are developing a thrombus and the shear increases, and then these receptors respond with more thrombus formation?
Yeah, it's hard to say about it, because that probably that just like the GPR56, it can be it can be self-activated at a very high shear rate. Yeah, probably that some receptors only work at a high shear rate. So really this,
and then the question from Dr. Alvarez would be, could it be that these shear sensitive receptors are located in specific vessels in the body only?
Yeah, probably, but we don't find it. Yeah, we just find that these receptors are everywhere.
Okay. I thank you for your answers and give the word back to the pro-rector.
Thank you.
Thank you, Professor Mariman. The opposition will be continued by Professor Ulrich Walter. He is a senior professor of translational research in thrombosis and hemostasis at the Johannes Gutenberg University in Mainz, Germany. Professor Walter.
Thank you, dear pro-rector, dear candidate. Let me also start by saying I'm very impressed by your presentation today. I'm very impressed by your thesis and the papers leading to this thesis. So congratulations for this. And of course, congratulations also to your promoters, which all brought it together. This is some nice work. Coming to the first question of mine, I'll go again, like other questioners here also, to the chapter 3, which I think is not only a major chapter, but also a major paper, and which brought together a lot of work, which I'm, again, extremely impressed about the content, because about the proteome and transcriptome of human and mouse platelets. So this is a very important paper for the field, I can tell you that, and I think will be cited in the future a lot. But I have a question to this. I think you spent some time with your colleagues and groups to estimate the proteome size of human platelets and compare it to mouse platelets. And as far as I remember now, you came up here to a size of 5,400 or 5,500 or something like that. So that's a decent number, and it's of course higher than previous publication, because things were improved by methods and so forth, and all the data together. So what do you make out of this? 5,500 proteins in platelets. As you mentioned, platelets are a very, very small cell. Some people even say this is not a cell because they don't have a nucleus, but I think it is a cell. But how do you compare this number of the smallest, or one of the smallest human cells, human platelets, to the human proteome? Do you have some idea about the human proteome altogether? What people discuss now is the size of the human proteome.
Okay. Hi, esteemed opponent. Thank you very much for your word and for your questions. Yes. I think the platelets are cells. Although it's very smaller. We identify in our database. We combine the 6 publications and get a database that contains 5,000 unique platelet proteins. And then we perform the new experiment and get around 5,100 platelet proteins. But when we compare this data to the human cell proteome, we find that numbers are smaller. Much smaller, because that in human proteome that we can identify more than probably 10,000 proteins. It's much bigger, but I think probably due to the the nuclear proteins, because that that the platelets are don't have the nuclear proteins and it's smaller. So yeah, so I think probably that many proteins are not in the platelet because that in the UniProt that there are more than 2,000 human proteins, but in our prediction models, we only find that around 10,000 platelet proteins. Probably that it means the platelets are indeed have less proteins compared to the
I mean, of course, it's still a huge number. I mean, that is 5,000 is a huge number. Okay.
Yeah.
The human proteome, to my knowledge, in the database and publication really studied it well, is about 23,000 proteins in the proteome. Okay. But does it really all define the function of the proteins? I mean, just the protein, there must be more than that. What could vary the function of the protein in addition to just being there? I'm talking about covalent modifications of the protein. What kind of modifications one could have in these proteins, which would make the protein function in the cell and other environments?
Okay, yes, for the modifications, I think that should be the phosphorylation. Yeah, we also focused on this part and get some data about the phosphopeptides.
Mm-hmm.
Yes, because we know the role of the platelet and it involved in the thrombus formation and the hemostasis, and the phosphorylation could regulate the platelet to complete its functions. So I think that probably we can, yes, the phosphor, we can pay more attention on the phosphorylation research about the platelet.
Mm-hmm. I think for the most important modification, phosphorylation, dephosphorylation, but there are also other ones. Can you just name a couple of other modifications, how the protein could be modified, could be degraded, or could be affected by its function?
Yes, because like the evacuation that it can affect the life of the cells. But yeah, we didn't focus on it. I just know these modifications.
Yeah, I mean, for example, by some other modifications, acetylation or ubiquitinations or other ones. So there are multiple modifications which are complicated to study, but they can modify the protein. But I think your basis is now a very important piece of work for future analysis. And I'll come now maybe to the second part of my question is coming to a major group of protein function, protein kinases. And you studied one, the focal adhesion kinase. And also, you also mentioned in the paper, this is a less well studied and understood protein kinase compared to other ones. Just briefly summarize to me and us here, what do we know really about the focal adhesion kinase in platelets?
Okay, yes, there are so many kinases in platelet, like the SAC and also the SHP1 and SHP2. Yeah, we were interested in the thesis. And I think the majority of the platelet functions are regulated by the phosphorylation and the dephosphorylation, like the protein kinase. And like if we inhibit the SAC, it's a kinase that the platelet cannot be activated by the GP6. So it means that it plays important role for the platelet or its functions. Yeah.
Just one question. I mean, most kinases are defined as their kinase activity. they phosphorylate other proteins or even themselves, often to regulate other proteins. And one important substrate of the focal adhesion kinase is SARC, also in platelets. So what would SARC, what would this do? I mean, if you phosphorylate SARC by focal adhesion kinase?
You may give a very short answer, please.
Very short.
Very short.
Okay, yeah. So for the first, I think we should combine the different experiments together. First, we find that which agonists or inhibitors could regulate the platelet functions, and then we can do the phosphoproteome research and to find which proteins are regulated and find the like the SAC, and we find that it has the important roles that it can connect with the SHP.
We have to move on to the next opponent.
Thank you.
Okay, sorry.
Thank you.
Thank you very much, Professor Walter. The next opponent is Professor Grüneboom. She is Professor of Experimental Biomedical Imaging, and she is affiliated with the Leibniz Institut für Analytische Wissenschaften and with the University Hospital in Essen. Professor Grüneboom.
Dear candidate, like my colleagues, first of all, I would like to congratulate you for to this very impressive work, this comprehensive thesis, these numerous papers that were successfully published, and also to this very clear presentation. We already heard some discussions on the impact of the shear stress on the platelets, and I would like to start a little excursus on this part of your work. Yeah. So you mentioned, for example, that the high shear rates are very important to modulate the GPR56, but for example, it's also very important in the CD62P, the P-selectin expression. And I hope that you are familiar with the aspect that the CD62P binds to the P-selectin glycoprotein ligand 1, the PSGL1, on the surface of neutrophil granulocytes. Have you ever heard of these cell types?
I'm so sorry, I didn't heard about it. Sorry.
Oh, okay. Well, then maybe let me introduce to you a little bit.
Okay.
About these cells. So neutrophil granulocytes are the main common white blood cells that we have in our bodies, and they are very important in defense of bacteria and so on. And they are now in the focus with respect to the interaction with platelets and thrombus formation. And well, maybe then this is now a bit more speculative since you haven't heard of these cells. So what's very interesting is that in contact with activated platelets, neutrophil granulocytes undergo a specific form of cell death, the so-called net formation. They form neutrophil extracellular traps. Maybe you have heard of these stuff. These are DNA fibers that these cells spill out to the vasculature. Have you ever heard of nets and net formation?
Yeah, I just heard a little about it, but I can't say that I know much about it. I just heard about it. Sorry.
Okay, so yeah, maybe then this is more than a transfer discussion to you. So when the nets are released, the DNA disposes histones, serine proteins, proteases, the tissue factor on the DNA. So could you speculate how this might affect the platelet function and the signaling in the platelets when they get into contact with this free DNA in the circulation?
Okay, yeah. Hi, esteemed opponents. Thank you for your questions. Yeah. First, I need to say sorry that I know very little about what you are seeing, but I think that probably it can be I can just give some answers. Probably that for the fibers and also the tissue factors, you said that probably we can just detect it by the flow chambers and to see the yeah, because I remembered that there will have the fiber generation at the very low share rate. Probably we can just do experiment like it at a very low share rate to see what will change.
Yeah. Okay. Thank you. So, for example, these negatively charged DNA in nets, they provide a scaffold for the factor 12 activation. And so could you also maybe speculate on that, how this then interferes with the platelets?
Yeah, to be honest, I know very little about the DNA, but probably we can try some experiments and flow to see that if it could regulate the platelets, like if it can activate the platelet to trigger the thrombus formation, or the other things that probably we can try. That's a good idea, but yeah, I'm sorry, I know little about it.
That's a very good idea. So this co-cultivation of the neutrophils or the nets with the platelets, this is something that some immunologists are also now addressing. So this is exactly what I would also recommend to do as a next step if you want to go more to the immunological aspect and pay attention to these cells. So this is absolutely right. And so would you also have a strategy then how to link the proteomics data that you can generate based on neutrophils or neutrophil extracellular traps and the activated or affected modulated platelets? Do you also have strategies to correlate these different types of proteomics data?
Yes, I think we can. So first, we should design the experiment to check if the neutrophil works for the platelet or the thrombus formation, and if we find it works, probably we can just collect the samples and to do the phosphoproteome analysis and the protein analysis to see the changes inside, and probably we can find something interesting, and probably we can do more experiment to validate it.
So with this, all my questions are answered. And yes, I would be very interested if you stay in science, if you might then also focus on neutrophils, and we could team up on that. Thank you very much.
Thank you.
And with this, I would like to give back the word to back to the pro-rector. Thank you.
Thank you.
Thank you, Professor Gruneboom. The opposition will be continued by Dr. Salgado Castro. He is University Lecturer of Biochemistry and Molecular Biology at the University of Santiago de Compostela in Spain. Dr. Salgado.
Thank you very much, dear Prorector, for the introduction. I hope you can listen me perfectly. First of all, I would like to send my warm compliments to you and your directors and co-directors, because this has been a really nice piece of work. I have enjoyed a lot reading it. So it's a lot of effort put in this work. So I would like to first of all recognize it. So I hope you will stay in science in the future, and I wish you a lot of success for it. So let's start with the first question. So that's regarding the stability of the platelet proteome transcriptome and some pre-analytical variables. So I would like to know if, so you have said, for example, that the stability of the platelet proteome is quite high. So it could be influenced by the sex of the age. What do you think? What do you think?
Okay, yeah. Highly esteemed opponent, thank you for your questions. And I think that the sex or the age could affect the platelet. Yes, it should be better. We didn't consider it here, but we use a lot of donors, like for the validation data. We collect the 30 subjects. That means that we can just combine this thing together. We can just ignore the effect of the gender or the age. But probably we can do more to separate we can do more about this effect to just compare the influence of the age or the gender. But yeah, probably we can do more, but we didn't do it. And I know that the aging platelet probably they may more active to the organism, and I think that it will change. But
I mean, just we are trying to speculate just a little bit. Just I know that it's really impossible to do all the work and in just 3, 4, 5 years. That's impossible. And you have put a lot of effort in this thesis, so no question about that. So let's speculate a little bit more. So for example, platelets are influenced by serotonin. So serotonin is a kind of activation factor for platelets. And did you know the connection between serotonin and melatonin? So would you expect some variation in proteome and transcriptome along the day?
Sorry, to be honest, I didn't heard this organism, but I think that probably we can just use this organism to treat the platelets, and then to see the changes of the transcriptome and the proteome, and then to find the relations between the transcriptome and the proteome, and we can find the difference between it. And I didn't do it, but I think that's probably we can do it. We can find something interesting in future by the transcriptome and the proteome.
Okay, thank you very much. So, and along the year, did you know something about the circannual changes in proteome? Do you hear about that?
Sorry. No.
Yes, the changes in the proteome that you could have along the year. Okay, you could have some variation. So what do you expect for platelets? Do you expect some variation in proteome platelets if you withdraw the blood, for example, in winter time, and regarding summertime, for example? What would you expect?
I think that the platelets, there are a lot of different in the single platelet, because it's that's the platelets are releasing from megakaryocyte, and the one megakaryocyte could release more than 1,000 platelets. So these platelets can be different. But we didn't. Probably we can just do like the single cell proteomics, or single cell the transcriptomics to see the difference. But we didn't do it. Probably we can find the. We can find.
I know. No, it's just thinking about the future, about the possibility for doing new things, for example. So you are for example, would you like to do single cell proteomics on platelets or single cell transcriptomics?
I think it's interesting, because I really want to do it, because like that we know that there are so many layers of the thrombus. So I think that the outer layer is different with the inside layer. So probably the single cell proteomics could solve this problem. So if I have a chance, I want to do it.
Yeah, but they are really small cells, piece of cells. So it will be really difficult to detect the proteins in just one platelet. It's not like a lymphocyte, for example, which is bigger.
I know.
And what about metabolomics? Would you like to do that? Or in the future, let's say, just to complete the map?
Yes, I think that it's interesting, because that there are a lot of research focused on the omics, that like the genomics, proteomics, and the metabolism mix. So I think it's better for me to do to combine everything together to find the difference between the different treatment or between the health and the disease. I think I hope I can do it in future.
So moving to another issue, what about the contamination of the platelet protein with proteins coming from the plasma? What is it big? Is it low? What in your work?
I think it's low because we wash the platelets for so many times, and we can make sure that the purity more than 99%. But it's so hard to remove all the contaminant like that. We detect the fibrinogen platelet, but it should be in the plasma. It's so hard to remove all the contaminant because we need to balance the samples and the contaminations. So yeah, we just choose that we do what we can do.
Hora Ast.
You will have noticed, Mrs. Huang, that the time appointed for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and the quality of your defense. And please await our return with the results of our deliberation.
Okay, thank you.
I see that we all are back now. Mrs. Jingnan Huang, the degree committee here present online has assessed the quality of your thesis and in particular the quality of your defense. And in view of its positive verdict, and of course taking into account your previous qualifications, the degree committee has decided to grant you the degree of a doctor. Professor Heemskerk is authorized to confer upon you this academic distinction in accordance with Dutch and Spanish university custom and law. May I invite your supervisor now to take the floor, Professor Heemskerk.
First, you'll get the promise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes.
Then, by the authority vested in us by law and in the conformity with the decision of the committee here present online, I hereby confer upon you, Jingnan Huang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, secretary, and the supervisor, affixed with the official seal of the university, shown at present by the beadle.
Yeah.
So this shows the signs of the two universities, the University of Maastricht and the University of Santiago de Compostela.
Thank you. Thank you.
Dear Dr. Huang, dear Jingnan. So you are a double doctor now.
Yeah.
Sing a little bit that you realize that you are a double doctor. It is a really honor for me to be the first person to congratulate you with this double doctorate degree from the Universities of Maastricht and the University of Santiago de Compostela. And I'm very happy also to make these congratulations on behalf of your other supervisors, Dr. Angel Garcia from the University of Santiago de Compostela, Professor Albert Siegmann from the Leibniz Institute ISAS in Dortmund, and Professor Hugo de Kerte from Maastricht University. And this big and special congratulations for a double degree also include, of course, your family and friends. So, sometimes it's difficult to give a laudation. For instance, if the new doctor did a master project in the same lab and then continued with the same topic and then just did well in 5 years of research, so then it would result in a laudatio full of platitudes, well done, made good progress and development, and some anecdotes on what happened in the lab or on a congress. But this is not the case now. So how much different is this in your case? It's now very easy to make a laudatio without platitudes because there is so much to say about you and so much to praise. But I have only a few minutes, so I must limit myself. In less than 4 years, you managed to write a thesis based on your scientific results of more than 400 pages. And now we spent some time to reduce the number of pages because in the beginning it was even longer, but now it's almost 400 pages. And then we need to realize that you had a kind of slow start in the first year related to your move from China to Europe. Your work has suffered from multiple lab closures and multiple quarantines that you had to undergo during the COVID pandemic. And you needed to divide yourself in working at the ISAS Institute in Dortmund, at Maastricht University, and at CIMIS in the University of Compostela, according to a quite strict schedule. And in between, you also needed to keep contact with your family in China. So there was a lot of moving around and a lot of changes that you had to undergo. And in addition, you needed to get knowledge of the thrombosis and hemostasis field. You have a very strong background in proteomics, but you need to learn a lot about platelets. And importantly, you need to adapt to the different environments of 3 European countries. All of them also so very much different from what you were used in China. So we can read in your thesis that you have been lucky to meet quite some other PhD students also in Europe, which probably helped you also to pass this period. Nevertheless, you managed in doing all of this in 4.5 years with a fantastic result. And then obvious, not only the length of the thesis is important, but even more important is the quality of the thesis. And regarding this, you have done quite well. Up to now, you have published or co-published 5 papers of your PhD research, all in good journals, and there are still 2 more papers to come. And in addition, during your PhD time, you are also co-authors of 3 papers from your previous home lab at the University School of Medicine in Shanghai, China. So even that continued while you were here in Europe. Now let's briefly go back to 2018, when you came over from Shanghai to join the Horizon 2020 TAPAS program. Your project title was Innovative Methods to Study Proteomic Signatures of Glycoprotein Receptor Signaling. So the idea was that you would do this by using new cross-linking methods, new proteomic methods, and then studying small molecule inhibitors. So at that time, we were looking for an analytical chemist with some biological background. And respectively, quite luckily, we found you, because you had both a biochemical and an analytical plus proteomic backgrounds. And what then happened, as it always goes with the PhD project, your own background and the circumstances, including the COVID lockdown, led to a redefinition of the project, with this thesis as a result. So it started with a nice review on platelet proteomics. Then it was followed by impressive efforts to define the complete proteome of human and mouse platelets based on transcriptome data. And once you could enter the lab, then you developed into a highly appreciated colleague in the platelet groups by working on several functional studies together with other PhD students. You are a very good collaborator, which also appears from the fact that on the thesis-related papers, you have as many as 40 co-authors. So it's an impressive high amount. At the same time, you defined your own functional studies, also linked to clinical relevance. And the aim in the end was changed by not asking how glycoprotein signaling occurs, but also how this is dampened, which is a new way of looking at the platelet functional studies. Regarding your own development, Jingnan, what I have seen in the past 4 years is a person who has grown from a fairly dependent PhD student to an independent researcher. And during that process, you have shown a big motivation to continue and move on, also in more difficult times. And in meetings, your standard remark always was, yes, I will do it. And then indeed you did it, and almost always with very good results. So I think you also had a good support in your research with the other participants and ESRs of the TAPAS program. This TAPAS program on platelet receptors, coordinated by Professor Steve Watson in Birmingham, has motivated all of us on working together on multiple aspects of platelet receptors and platelet functions. And in the contents of TAPAS, there were multiple meetings for interaction between the PhD students, which probably will help you a lot during the course of your project. The pity still was that the anticipated meeting in Santiago de Compostela in the end needed to be replaced by virtual sessions. On behalf of the other supervisors, Professor Albert Ziekman, Dr. Angel Garcia, and Professor Hugo de Kaete, I thank the members of the assessment committee for their work and their presence today. Dr. Ezequiel Alvarez Castro, we feel very sorry for the small accident and wish you a quick recovery, and thanks you for your efforts. Dr. Francisco Salgado Castro, also from the University of Santiago de Compostela. Thanks for your presence and questions Professor Annika Groeneboom from the Leibniz Institute Dortmund, and Professor Ulrich Walter from the Johannes Gutenberg University in Mainz. Thank you as well for your presence and questions today. And last but not least, Professor Edwin Marreman and Professor Monica Stoll, representing Maastricht University. Also, thank you for your valuable participation in the thesis, examination, and defense. And dear Jingnan, now, since the last summer, you are back in your proteomics lab in China, where you surely will be successful as well. And before handing over to Paul Rector, the last thing for me is to show you the presents collected in the lab. I have those in front of me. I will show those at a later stage. And I will hand those over in a symbolic way to you. And those will be sent to China as soon as possible. And with this, I hand over to the provider.
Thank you.
Esteemed Dr. Jingnan Huang. Dear Jingnan, it's my great pleasure to congratulate you with your double doctorate degree of the University of Maastricht and University of Santiago de Compostela. And I do that also on behalf of the board of deans of both universities. And maybe I may share some impressions of the committee with you.
Okay, thank you.
To start with your presentation, we enjoyed it. We consider this as very good, very clear, and we are aware of the fact that it is quite difficult to summarize all the data in less than 15 minutes. So that was very clear, very good. We appreciated that. And we have seen also a very impressive thesis. Well written, comprehensive, with lots of data. And we consider this as very good work. And also we have seen that most of the papers are published in good journals. So we appreciate that as well. Your defense, we have seen that you are really majorly involved in the field of research. And we also have seen a good background knowledge. Of course, we are aware of the fact that you did not answer all our questions, but the most questions were very well answered and very adequately. And to take it all together, we also appreciate that you manage your studies in quite difficult circumstances and conditions in Germany, in Spain, in the Netherlands, and in the United Kingdom. And we consider that as a major achievement. And I congratulate not only you with that, but also your 4 supervisors, Professor Heemskerk, Professor Siegman, Professor Dr. Garcia, and Professor Tenkate. Congratulations with the results that we have seen in the form of an excellent thesis and a very good defense. Congratulations with that. And of course, I would like to include in my congratulations, Jingnan, also your family and all, not only your parents, but all the members of the family that have been involved in educating you and let you leave to Europe. And we have seen now that you are really in a family of well-respected members. And I would like to congratulate not only your family members, but also all the colleagues and friends and all those that have been involved in coming to all your studies and in the form of thesis and defense that we have seen today. Before I close this academic ceremony, I would like to thank all the members of both the assessment committee and this degree committee, and in particular the external members and all opponents for their questions and interesting academic discussions. Maastricht University appreciates your contributions. And now we have come to an end of this academic ceremony, and hereby I close this ceremony.